Hello again, Dr. Dobbs McAuliffe here. We're going to continue with the last chapter in my section of the course, which is chapter 16, focusing on cell signaling. And this is going to be how cells communicate with one another. We're going to begin with general principles of cell signaling, and then look at two common families of signaling receptors, the G protein coupled receptors and the enzyme coupled receptors. In cell-cell communications, cells often switch signals from one form to another. We just looked at an example of this in neural signaling, where we converted signals from an electrical signal, which was an action potential, to a chemical signal, which was a neurotransmitter. The text presents this analogy of your cell phone, where a radio signal is what comes into the phone and is converted, or you might use the word transduce, to the sound of a person's voice. That's the output. So the input is the radio signal and the output is the sound. Of course, there's intermediate steps between the input and the output. In the picture here, they're trying to show you a similar situation where we have input, which is a signaling molecule. This is going to be received by a receptor. Here they're showing you the output is an intracellular signaling molecule. I'd say that, to me, that is more like the inner workings of your cell phone. For me, the output would be the change in cellular behavior, which might be something like cell movement or transcribing a new gene or altering the cell's metabolism. Cell signals can be classified by the distance between sending and receiving cells and how the signals travel. This first example is endocrine cell signaling. And these are signals with familiar names, things you've heard of before, like estrogen, testosterone, cortisol, adrenaline. In this case, the cell producing the signal, the endocrine cell, can be at a great distance from the cells that receive the message. And the message, or hormone in this case, can travel through the blood to various target cells. Only cells with the appropriate receptor, so a receptor that binds to the particular hormone in this case, will respond to the signal. This allows endocrine signals to coordinate activities in multiple organs. For example, adrenaline from the adrenal glands on top of the kidneys can affect heart rate and blood flow to the skeletal muscles and brain. When the cell producing the signal is nearby the target cell, only 1 or 2 cell diameters away, this is paracrine signaling. The simplest way that paracrine signaling works is for the signaling cell, the cell producing the message, to produce the signal in vesicles, which then fuse, are released into the extracellular space, and then can diffuse to nearby cells. Again, only cells with the correct receptor will respond to the message. This is the most common type of signal in developmental processes, such as regulating cell division, movement, differentiation, morphogenesis, which is kind of creating the shape of particular cell types, and this is happening as embryos are growing. So the third type is neuronal signaling, which we just covered in chapter 12. In this case, the cell bodies of the two neurons can be quite far apart, as much as a meter or so. However, the axon of the presynaptic neuron is going to be very close to the cell body or dendrites of the postsynaptic neuron. There's a small space called the synapse that separates the end of the presynaptic neuron that's producing the signal and the membrane of the postsynaptic neuron that's receiving the signal. A fourth way to characterize cell signaling by the distance between the cells and the way it travels is contact dependent. In this case, the cells must be directly adjacent or next to each other, because not only is the receptor stuck in the cell membrane, but so is the signal. I'm a developmental biologist, so I also often think of examples in developmental biology. When I see this type of contact-dependent signaling in embryonic development, this is often used to make repeated patterns of cell types and/or boundaries between different cell types. Signals can also vary in the location of the receptor. In the case of extracellular signaling molecules, these are signals that are too large or charged or both, and therefore cannot pass between the phospholipids of this phospholipid bilayer, the plasma membrane, and instead must bind to a receptor that is found in the cell membrane. Smaller signals that are hydrophobic, they have no charge, can cross between phospholipids and enter the cell, and they bind to an intracellular receptor. This might be found within the cytoplasm, or in this case, the receptor is actually within the nucleus. We're also going to look at the way the effect of one particular signal can vary between different cell types. So I'm going to show you 3 different cell types. In each case, the signal is acetylcholine, but the effect on the cell will be different. So acetylcholine has different effects in different cell types. It can vary in the intracellular signals and effectors, and also on the receptor type that is used to detect the signal. First, we have a heart pacemaker cell, a cell that's going to regulate the speed at which the heart is contracting. We can see the receptor protein here. Here's the signal acetylcholine, and we're showing a decreased rate of firing of the cell. So if we look in this picture, we see the same type of receptor, but it's in a salivary gland cell. In this salivary gland cell, we have vesicles filled with the components of saliva. They look a lot like synaptic vesicles at the end of a nerve. These, instead, are vesicles containing the components that make saliva, and we can see that acetylcholine triggers the fusion of these vesicles and releasing those components. Acetylcholine can also affect skeletal muscle, but we can see that it's the receptor that varies here. There's a different type of receptor to which acetylcholine binds. The receptor is an ion channel. When we open the ion channel, the effect in the cell is contraction or shortening, which allows us to move our bones. So we can see that we have variation in the type of receptor between skeletal muscle and salivary gland cells, where salivary gland cells and heart pacemaker cells both have the same type of receptor. This is a G protein coupled receptor, something we're going to cover in more detail shortly. Rather than having one cell signal for every effect that can happen in the cell, signals are combined to cause different effects depending on what signals they combine with. So I'll try to go through some examples here. In this first cell, we're seeing it's receiving 3 different messages. So we're activating 3 signaling pathways, A, B, and C. These 3 signaling pathways are required for the cell to survive. We can then combine these survival signals, A, B, and C, with different contexts and see different effects, or we can remove them. So actually, let's start with the comparison between the top. The cell has A, B, and C signals active, and the cell is surviving, whereas if we remove these 3 signals, the cell will die through a process called apoptosis. So cells have to actively repress apoptosis pretty much at all times. If the cell is receiving A, B, and C, and is therefore surviving, it can also receive other combinations of signals along with the survival signal. In this case, we have signals D and E. So the cell is not merely just surviving. Instead, it is also growing and dividing. The third cell is receiving signals A, B, and C, which promote survival, but also signals F and G. So the outcome is different. Rather than growing and dividing, the cells are differentiating or becoming a very specific cell type. If you're not familiar with differentiating, some good examples of cell types to think about are neurons or muscle fibers, very highly specialized cell types. So if we're thinking about a neuron, the cells that are going to become neurons initially start off as these very roundish cells, but as they differentiate during development, they get the correct signals here, the signal F and the signal G, they will extend dendrites and extend an axon, and they will gain the ability to release neurotransmitters and affect the likelihood of a postsynaptic neuron firing, for example. So the cell's response to a signal can also be subdivided in the speed of that response. Here they're showing one signal that can produce both a quicker and a slower response, both to affect what they're saying is cytoplasmic machinery. So this is a little bit vague, but let's say, imagine we have, in this case, an enzyme that's in an inactive form, and the signaling pathway's job is to activate or change the conformation of that enzyme, and then the enzyme can do its work. So that happens fairly quickly, less than a second to minutes. At the same time, the signaling molecule can activate the receptor in a pathway that activates gene transcription, making RNA, and then translation and folding and all of that to get an enzyme that is produced. Even if you're thinking about the same enzyme, there's going to be a difference in speed when you have to just activate the enzyme or transcribe, translate, and fold that enzyme. This is a slower process that takes minutes to hours.